Me too.
Wait, so
Is the
what
link
are
up?
So at the moment, there's no.
Yeah. But they can see all again. They can see us.
So is that all?
Alright, let me, how do I? I'm marking this as announcement.
And people are joining. Yeah, mark this announcement. Hey, Lucy's here.
Notifications for this post.
Hey, look at all this. Look at everyone joining, guys.
Oh, sorry.
Alright.
How do I copy a link?
Is it? Can you see audio? Can you see audio?
Oh, we can see.
Can you?
Yeah, I can.
Can you hear it though?
Huh?
Can you hear audio though?
I can hear it. Yeah.
Okay.
Did this let me try?
Yeah.
That's good. That's good.
Wait, can you turn the volume up?
Am I doing it right?
Yes.
What are you doing?
I think you can hear it.
Am I?
Yeah, I think so. Maybe. Do you want to like just start interacting? Yeah, start interacting. Just Q&A.
Yeah, let's just do that.
Do you want to just have Q&A? Guys, come on. Hi, John. Hey, guys. Sorry, come here. Hey guys, so we'll be starting in like 8 minutes.
We'll just chat for a while.
Yeah, this is most of us. We're missing one because they weren't allowed to come out. Yeah. But yeah, let us know if you have any questions, guys.
No.
You think it's up in that?
I was looking at that and I was like, why is there such a big comment? And then I realized that you.
That was Dave.
I marked that as an announcement.
Yeah. Okay, so 20 seconds behind. Yeah, turn your sounds off.
Say hi to all the Jaffies.
Yeah, bro.
I love the Jaffies.
Hi, Jaffies.
Hello.
Big sad for missing one. Yeah, they were like, oh yeah, our parents won't let us come to Monash because of Corona.
Oh, Jating's positive.
Yeah, bro.
Where's Jating?
Yeah, Jating's the missing one.
Wait, why can't I see her comment? Oh, wait, because it's like lagging. Oh, alright.
Alright, so how to work is essentially we have like about 2 hours to rip through all the content. So that includes week 1 and 2, as well as some Q&A session, if we have time at the end. So hopefully we can rip through everything. So hopefully we can rip through everything in time. But yeah, does anyone have any questions about life, about week 1 or anything like that? Central dogma. Oh yeah, true. Yeah, we heard about your central dogma lecture. We also heard about your body fluids lecture last week with Chris Wright. So we'll be going through body fluids, so like your osmosis, your osmolarity, osmolality, and then in week 2 we'll also be going through central dogma with a few other things.
Where are they sending questions?
Enoch, you can send questions just on the chat.
Yeah, just I think because Jating's
on the chat, and then we'll post up a Google Forms at the end if you have any like questions that you think of after, and we'll answer them in a big Google Docs.
Yeah. Yeah, Chris Wright, man. Chris Wright's pretty good. He's a good lecturer. It's just technical difficulties.
He always wants to write out his like lectures, and sometimes it just doesn't work.
Unsustainable. Oh, what gold. Yeah.
What's my comment?
Yeah.
Pink.
Osmotic. Oh, he's going through. Are you going through? Yeah.
You know, I just realized osmosis is actually really relevant.
Yeah. Well, things are relevant.
Wow, physiology meta actually relevant.
Wait, hang on. Here's a question. Do I go to my hand washing and injecting practical, or am I advised to do that online?
Like, you mean like your actual assessment? Because please do turn up to that, like, or you'll fail. The tute.
I think stay home for the tute.
We'll upload a video. Like, I'll do a video.
Yeah.
And it's really easy.
Heidi will show you how to wash your hands the best.
There are tips and tricks, so you won't like get a
needle. So you won't stab yourself.
Yeah.
Where's the auto zoom? Sorry, not cool enough to have auto zoom. How? Oh yeah, so pinch this, just pinch your imaginary air in front of you and go at a 45-degree angle, holding like a pen, and just like aspirate and then push in.
Yeah, wait, med school hoodies. Sorry, Yakuza, you'll get this soon enough.
Oh yeah, your med hoodies. We should have come in our med hoodies.
Yeah, Zoe's not wearing hers.
Yeah.
Yeah.
I love my other hoodies. I've been wearing that literally all the time.
Yeah.
I need to put in the wash.
I should have worn my med hoodie. It was so cold this morning.
For real. March med hoodies. I feel like they might. Do you think they'll let ordering online happen?
Isn't it like a thing though? Like,
Yeah.
It's like on the Meiosis.
I'm sure you guys will get a hoodie. It'll come eventually. Last year they did a bit of like, you can order it online and then you can pick it up at a certain date. So hopefully they can do that with you guys. But yeah.
Hey G.
Welcome to my vlog.
Welcome to the vlog.
Live, yeah. Alright, about 3.5 minutes, yeah? Yeah. Oh, 70 people.
What
God, I'm so proud of this turnout.
For the semester.
Why did you tell me?
It's online.
You tell them.
But what is there to look forward to?
Oh, what is there to look forward to?
Even though like your GP visits are cancelled, if the hospital visits are still happening, which I don't know if they are, but those ones like later clinical exposure is like really fun, especially as an early approach to what it might be like to work as an intern later in the future. So hopefully there's a few more things faculty has prepared for you guys just to look forward to. I think that's pretty important. They will definitely make it up to you. Med faculty is really good at supporting you all, so it should be alright.
Yeah, for sure. What else is there to look forward to?
Well, I was gonna say like all the med events, but then they're all cancelled.
F in the chat. F in chat. F in chat.
Please join me Taekwondo club.
Yes, this is Zoe. Zoe is what do you hold in Taekwondo club?
I'm VP of Taekwondo club.
Yeah.
Please join.
Yeah, Taekwondo club.
They also need money.
10.
It'll help you fight off coronavirus.
Yeah, exactly.
Yeah. Exercise.
Like axe kick.
Yeah, just axe kicks the coronavirus away.
What else?
Medico premiering on YouTube soon. Oh.
For those of you who are super keen on getting a step, we've got like a discount code for later, so keep an eye out.
Please don't get me like you don't.
We've got a discount code later as well, so if you're really keen on getting one, just like message us privately and we'll send you the discount code.
But you don't need a step this term,
Yeah,
this semester.
you
You also do not. You don't need a lab coat. You don't need safety goggles. Ever.
You need a Wi-Fi connection.
Yep.
Yeah, you just need a Wi-Fi connection and a laptop. So yeah, hopefully Medball isn't cancelled. Yeah, ching. Oh, that would be so...
Bro, if Medball is cancelled, we'll just hold our own one.
Yeah, PS Pingu,
Yeah,
Medball.
PS Pingu,
Yeah.
Medball, everyone
Online. Yeah.
Online.
Online. It would just be like mukbang for everyone. Yeah, where's the mukbang, David? Where's the Neptune chips?
Yeah, sorry guys. Guys, I was gonna bake brownies for everyone. I was gonna buy chips as well, and I'm so devastated that we can't like actually do that. But
Yeah.
we'll do it for our first like.
Yeah, don't worry. There'll be food when you guys come here. Hopefully, if we're allowed to bring you guys back on campus. But who knows? You guys probably haven't been. Oh, some of you would have been in this building for interviews. So it's a bit of PTSD for you. But we're currently in the med building. This will be your faculty building. It's really nice, actually.
Basically, you spend like most of your life in here.
Yeah. There's hand sanitizer in here. Yeah, still got like 30 seconds.
Oh, mine's.
There's some hand sanitizer in here. There are bathrooms. There's a toaster press. There's a microwave. There's even a fridge. There's hot and cold water, free tea, coffee, Milo. Oh yeah, Milo is very popular though. What else is there? There are beanbags. There are beanbags in the med building.
Yeah, but they always get stolen. Like people like bring them into the rooms and stuff.
Yeah. Okay, dokes. So.
Watch party on Med 2024.
Holy shit. Okay.
Should we get started?
So we're gonna get started.
There's some introduction. We're gonna do
Yeah.
because
Alright, so you guys can follow on, if you would like, on our week 1 slides. That's on our Facebook group or on the Meiosis academic drive under year 1 PSP content. So feel free to follow along as we go, but you'll be able to see exactly the same things as we have. Okay, sweet. We'll probably start introducing. What do you want to start with?
We'll just start on our slides then.
Start with the slides?
Yeah, we can just go through like this.
you need to introduce everyone.
Alright.
Yeah.
Oh, this one's just even more laggy.
Oh, okay.
Yeah.
And then it's working.
Yeah,
It's working?
Alrighty. This PowerPoint we made a while ago without Corona. So the plan was to have weekly recaps, but at this point it looks like it'll probably be at 11 a.m. And in addition to 11 a.m., it's looking like it might be online for a few sessions. Yeah, quite possibly indefinitely. But we'll figure something out. If there's lockdown, we'll still figure a way out. We'll probably end up recording videos at home and uploading them for you guys. Facebook Live's already better than half-hour lectures. Moodle is questionable at times.
Just you have to learn to deal with it. Like
Yeah.
sometimes it works really well, and it's like awesome.
Yeah.
Yeah, but like Anyway.
Yeah. We'll give you the textbooks. That'll be on our Facebook page, but also do not upload anything. We'll give you a whole document with a bunch of resources. Also, if you want to get access to that drive, you have to be on your Monash email. That's why a lot of you probably can't get in. There are also some folders that are only for us because we're preparing stuff for you guys. Studying areas doesn't matter for now. Just use your laptop.
If don't skip too many lectures, you will regret it. It's
Yeah. Alrighty. So we got to move on because we actually have a lot to get through.
Okay.
So Heini will be starting off with cell anatomy. Have fun.
Okay, hi guys. I'm Heini. I'm going to be taking you guys through just really quickly going through cell anatomy. So for those who have done bio 3-4, you can kind of just like tune out for the time being because this will be stuff that you already know. So what we're going to quickly go through, we're just going to talk about cell theory. So basically cell theory, so the difference between original cell theory and modern cell theory is basically modern cell theory is the same as original cell theory. They just like figured some more stuff out and then just like added onto it. So you can see in the table here, some basic things that you just need to know is that all of life, so like all living things, are composed of one or more cells. So basically cells are the basic structural and functional unit of life. It's, you know, and all like cells are produced by the division of pre-existing cells. So basically cells come from other cells. And then in modern cell theory, you have a little bit extra. So they figured out that cells contain DNA. They have the same chemical composition and metabolic activity. And then like you can read the rest. Honestly, this is like a little bit boring, so we'll just go through it really quickly. Where do I? Oh, what the fuck? Oh my god. Okay, hand-eye coordination. Okay, so the difference between prokaryotic versus eukaryotic cells, it's pretty simple. Just like look at the table, and this is basically all the information you need to know. Some like key information, prokaryotic cells have circular DNA that's usually free-floating. Eukaryotic cells, on the other hand, have linear DNA, which is found in the nucleus. Basically, prokaryotic cells are usually smaller than eukaryotic cells. And then, yeah, honestly, like, we can skip this. Please just like look at the slides again if you want to learn any of these in detail. I put a lot of notes in the speaker notes as well, so you can have a look afterwards, but we'll just fly through these. Okay, so you need to know about the cell membrane. Basically, it's a phospholipid bilayer. So it's made of two phospholipids with their tails pointing inwards, as you can see in the picture up here. So it's selectively permeable, which means that it basically selects what they let through the cell membrane. So small molecules and non-polar molecules move straight across. However, polar molecules require transport proteins or protein channels. We'll go into a little bit more detail. Okay, so another thing that you do need to know is the fluid mosaic model. Basically, it's just what it means is that the plasma membrane can move freely and fluidly, and it's like constantly sliding against each other. And so basically that means that if you insert like a very, very fine needle into the cell, the membrane will actually flow around the needle, and once the needle is removed, the membrane will just flow back together. So it's very fluid, it's always moving. These are some key components of the cell membrane. I'm not going to go into detail, you can just like look on it yourself. A lot of this unfortunately is memorization, but it's pretty simple once you like understand the basic concepts. Here are some good websites just to learn a little bit more about cell anatomy, especially this first one, organelle histology, because you are going to be doing a lot of looking at a lot of histology slides, especially in active learning, and a lot of the time like you don't actually know what it like looks like. So like to just like, get a little bit more practice with like learning how to identify stuff. Okay, let's go into organelles. Okay, first one, most important, is the nucleus. So this contains the cell's genetic material, DNA. DNA is wrapped around histone proteins to form chromatin, and then you've got two different forms of chromatin, heterochromatin and euchromatin. Basically, the one that's active is euchromatin, so that's like the loose one, and that's the one that's like constantly going through like transcription and translation. And then chromatin is further coiled to make chromosomes. So some key parts of the nucleus, you've got a nuclear envelope, which basically is like it encloses the nucleoplasm, which is here. So it's basically like, this is like your cytoplasm of the cell, but like the nucleus. You've got your nuclear pores, which basically regulate the substances that are entering and exiting. And then you've got your nucleolus, which is where new ribosomes are synthesized. Okay, sorry if I'm going like really fast, but you can just like look back on the slides. Okay, mitochondria. It's the site of ATP synthesis, so basically where all of your energy comes from. It has a double membrane, and it's actually inherited through your mum. So like you can trace back like mitochondria back through like generations. It has its own DNA and ribosomes because it was actually originally a bacteria, which was later integrated with cells through endosymbiosis. So basically, like the cell just came along and just like ate the mitochondria and was like, okay, it's pretty cool, I'm gonna keep it, and that's like, that's it, yeah. Let's keep going because we're running out of time. Okay, ribosome is basically where protein synthesis happens. It is not an organelle because it is not membrane bound. However, it's really important. It's made up of a large unit and a small subunit, and that's kind of how it looks like, and the mRNA will sit between the two. So the mRNA comes to the ribosome for translation. The ribosomes can be free, which is just like floating in the cytoplasm, or they can be bound to the rough ER or the nuclear envelope. Keep going. Okay, the smooth endoplasmic reticulum is responsible for the synthesis and metabolism of fatty acids, lipids, and their derivatives. So, yeah. It's basically there's no ribosome. That's why it's smooth. What's the time? Oh God. Okay, so the rough endoplasmic reticulum. This is the one that a lot of people do know about. It's responsible for the production and modification of proteins. So the modified proteins will either be incorporated into the cell membrane or they'll be exported outside of the cell. It's actually continuous with the outer nuclear membrane, which you can see here. So it actually like continues on from the nuclear membrane. And the outer surface is studded with ribosomes. So that's the difference between smooth and rough, is just smooth doesn't have ribosomes, rough does. Okay, Golgi body, it's basically like the post office. So it basically sorts, processes, and packages products into like little vesicles over here. Oh, where are they? The vesicles here for transport in or out of the cell. And basically it receives the vesicles from the endoplasmic reticulum from this face, and then it basically like travels through here and goes through like modifications, and then it's like exported. Lysosome, basically it's an organelle containing digestive enzymes. It's mainly involved in like waste removal, organelle recycling. It'll break down unnecessary structures that don't need to be used anymore, so that these molecules can be reused in the future. And they're very abundant in phagocytic cells. How are we going for time? Good. Okay, so now we're moving on to the cytoskeleton. So basically it's like the inner framework inside the cell that allows it to keep its shape and its structural integrity. So the main functions that it serves is that it helps organelles like move around inside the cell. It helps the cell itself actually maintain shape, and it actually helps the cell move around in the body. So the major components that you guys need to know about are microtubules, microfilaments, and intermediate filaments. Okay, so basically this is a table. This is kind of like the summary of like what you need to know. But like I'll just skip through that so we can go into more detail. Okay, so microtubules, they help maintain cell shape and they control transportation of cell substances in and out of the cell. So it basically helps like the cell like travel around, which you can see with cilia or flagella. I think there's a slide next. And basically in mitosis, the centriole is formed by 9 groups of microtubules, which you can see. I think it's this one. And the microtubules assemble into what's called the mitotic spindle, which helps actually pull chromosomes apart. We're going to go through cell cycle in a bit, so you'll be able to. Yeah, so this is the structure, the 9-2 arrangement for cilia, and basically just helps cells move along. And the flagella is the same structure as this, it's just like a longer, it's like the tail. Yeah, okay, we'll go through this in cell cycle. Okay, microfilaments, so these are 2 different types. So there's actin and myosin. Basically, actin, there's a lot of it. It's arranged in a double helix, and it's very important for like movement. And then we have myosin, which is basically you don't really see it that much except in muscle cells, and you can't see it under the microscope in normal cells. Arrangement, so basically you've got skeletal muscle, and then you've got the actin sheath beneath plasma membranes, which helps with cell movement and any like transportation of substances. Honestly, like let's just skip. Sorry, I want to get this like done. Just like read the slides if you like need more information. So okay, last one, intermediate filaments. Basically, they're made of multiple strands of fibrous proteins that are wound together. They also help maintain cell shape, and it kind of like anchors the nucleus and all the organelles, so it's like they like stick in their kind of places. And they come in a number of different varieties. Here are some examples. You don't need to memorize this, like just know what they are and know what they do. Please don't like waste time trying to memorize like all these different examples of stuff. Just like know the basics and then move on. Okay, cell cycle, go.
So we'll be doing cell cycle now. Once again, this topic is covered in Bio 1-2, so it's probably a couple of years since you guys covered it, but we'll also be ripping through this fairly quickly, so we can get to Chris Wright's body unsustainable body fluids, because I hear that was pretty troublesome. So to get started, there are two types of cell proliferation, mitosis and meiosis. Mitosis is essentially for your somatic cells, whereas meiosis is for your gametes, or your reproductive cells. Cell theory basically states that all cells must be derived from another cell, so it always replicates. So we'll start off with the entire cell cycle for now. And this is made of a few stages, so it's made of interphase, mitosis, and cytokinesis. And we'll be looking more into interphase and more into mitosis later on in this PSP. So essentially, your cell will go round and round this cycle, and that's how it replicates. There's also a G0 stage, which we'll be talking about in here. So we'll start off with G0. G0 is essentially when the cell isn't replicating, so it's just in rest, and they never leave that stage. But then coming back to the cells that are replicating, we start off in G1, and G1 is the longest period of the cell cycle, and it occurs so the cellular contents can be multiplied, and the cell can get ready for division later on. After that, we move on to the S stage, or the synthesis stage, and this is where DNA replication occurs. And we'll be talking about the actual process of DNA replication later on in this PSP as well. I think that's under the central dogma. So this morning's lecture, so I'll be spending quite a bit of time on that. But essentially you double your chromosomes, and then we move on to the G2 phase, and this prepares for mitosis. So once again, it undergoes a few more checks, checks for DNA replication errors, and the cell grows a bit more before finally splitting in mitosis. Now in mitosis, oh no, sorry, this isn't the slide. So basically the whole goal of mitosis, or the whole cell cycle, is to grow and replicate. Yeah, pretty self-explanatory. So if you cut yourself, you'll be able to heal back in time. Continuing about mitosis and cytokinesis though, mitosis is the division of the cells, division of the nucleus of the cells, whereas cytokinesis is the division of the entire cell. So mitosis occurs before cytokinesis, and it consists of 4 stages. So prophase, metaphase, anaphase, and telophase, or PMAT if you prefer acronyms. So if we start off with prophase, we can split it up into 2 sessions, into 2 kind of sections. So in early prophase, your chromatin or your DNA starts to condense into a heterochromatin. And then in late prophase, you start to lose your nucleolus. Spindle fibers, as Heini was saying before, will begin to form, and your chromosomes are fully condensed at this stage. Then we come into metaphase. This will be the stage where your chromosomes are most visible or the most condensed. And this is where we typically get our karyotypes at this stage here. They also align up in the middle. So you can think of metaphase starting with M and middle starting with M as well. Your spindles also grow and attach to the centromeres at this point. And then following metaphase, we get into anaphase. And anaphase goes for away, so your spindles will contract, pulling the chromosomes apart, and your sister chromatids will move to opposite poles of the cell. Now coming back to telophase, after we kind of have 2 groups of chromosomes, we'll have our nuclear envelope reassembling around these chromosomes, and the cell begins to pinch in to cleave the 2 cells into 2. In addition to that, our chromosomes also decondense into chromatin again, or into euchromatin. And after this, you basically finish mitosis, and the cell cycle will start again to keep replicating. So we just talked about mitosis there. Meiosis is also another form of cell replication used for gametes, or your reproductive cells. It's important to know meiosis, however, it is not as important as mitosis, I would say. It is essentially mitosis done twice. And the main differences are that meiosis produces 4 cells, and the DNA content is half of that produced by mitosis. So yeah, meiosis we'll probably skip through very quickly, but we'll go through it. So as you can see here, interphase, and then they pair up. Recombination occurs to get your genetic diversity. Your spindles fire out and attach to the chromatids, they're pulled away once again in anaphase, and the cell pinches off in the middle to form two cells. So that's the first part of meiosis, and then we'll go into the second part of meiosis. So in this stage, it's essentially the same. Once again, your spindles grow out and attach to these DNA in the middle. You can also see that the DNA in the middle has a bit of mix, so dark blue and light blue here, and light blue and dark blue. That's due to the recombination we had in prophase 1. So that contributes to our genetic diversity. Anaphase, once again, they're pulled away, and at the end of the day, we get 4 daughter cells, or 4 granddaughter cells, if you compare it to the original first cell. An important thing to remember is that these all are N, so it's half the normal genetic content as you normally would have. The reason for that is because if you combine 2 of these cells together during fertilization and reproduction, you'll end up with a cell with a normal number of, with a normal amount of DNA. If this doesn't happen, we run into a few disorders, which we'll go through later on. So we kind of went through prophase, metaphase, things like that. Once again, I'm skipping through this not because it isn't important, it's just because we want to spend a bit more time on other topics that are a bit more important and give you guys some time to ask questions, because I think that's the most useful for you guys. An important point to note, however, is that there's no interphase between meiosis 1 and 2. The reason for that is because it would completely disrupt the whole process, because we're trying to reduce the amount of DNA, and going through interphase again would increase the amount of DNA in the middle of it. Another difference at the end that I'll shortly touch on is that for meiosis we have a production of 4 daughter cells. However, this is different between males and females. So for males, during meiosis, we produce 4 sperm cells. However, for females, you'll be producing 1 egg cell and 3 polar bodies. The reason for that is because the egg has to contain a lot more information, has to contain all the organelles, all the information required for embryo development. So it has to be larger. So more resources are put into that 1 egg cell. However, overall, if you count the polar bodies as well, you still have 4 cells being produced. Genetic variation, I've surely mentioned it before with crossing over. So crossing over, once again, occurs in prophase 1, and it's essentially where 2 homologous chromosomes will essentially exchange a bit of their DNA information. The other sort of genetic variation we get is called independent assortment, and this occurs during meiosis 1, during metaphase 1. And we have different alignments and different arrangements of how homologous chromosomes will line up across the middle plate. So depending on how they line up, you'll get a different daughter cell. And then lastly, random fertilization isn't really concerned with the actual cell cycle itself, but it's essentially saying that during reproduction, you have millions, if not millions of sperm cells, millions of egg cells, and it's essentially random between those 2. So that's how you get some genetic variation. Okay, so we sped through that, but as a quick recap before we go through this, the most important bit to consider is during metaphase 1 and 2, your chromosomes are going to be split apart and pulled apart by your spindles. The important part to consider is that it has to occur evenly, otherwise you're going to get uneven and abnormal gamete cells at the end of the day. So if this doesn't happen, we call this non-disjunction, and that's what happens when chromosomes don't split properly. So if you look at this diagram here, if we have this non-disjunction occurring at meiosis 1, if we have non-disjunction occurring at meiosis 1, that will affect all the cells. So both these first 2 cells won't have any DNA content, but these 3rd and 4th cells will have double the amount of content. Secondly, if it occurs in meiosis 2, it's less severe because only half of the cells are affected. So our 3rd and 4th cell here will have the proper amount of DNA, whereas these first 2 won't have the proper amount of DNA. And typically it results in a spontaneous miscarriage because it's not developmentally viable. However, we do have some exceptions to that, such as these ones here. So we have Down syndrome, Edwards syndromes, and Patau syndrome, as well as Turner and Klinefelter. These are simply just a list to know. During ICLs or your Friday classes, which you guys should be starting this week, you'll be going through Down syndrome, and that's essentially trisomy, so trisomy of 21. So trisomy indicates 3 when you should really have 2. So these are the ones that can generally get through, but you don't need to really know too much about Edwards, Patau, Turner, or Klinefelter syndrome. Down syndrome is the main one that you need to know. Okay, so before we were talking about these large stages, so G1, S, G2, mitosis, and cytokinesis. However, between these stages, it's also important to have checkpoints. It's important to have checkpoints to ensure that the cell is replicating properly and so that we don't run into any issues. So to try to avoid any situations of non-disjunction. It can also cause issues during your life, such as cancer, because that's an accumulation of various DNA errors. So in our cycle, we have 2 types of regulators. They're both cyclins, so cyclins and cyclin-dependent kinases, and they work together. So these 2 cyclins and CDKs will combine together and it will attach to a target protein. And essentially, after activating this target protein, it'll cause regulation of the cell cycle. This content here is not too high yield, however. However, you do have to know the names of them. I think that's useful to know. It's kind of simply a list, unless anyone has a way to remember the cyclins. But I don't think it hasn't been very relevant for us. Yeah, it's essentially, if you want to try form words with it, it's DEAB, and then 4221. Not too helpful, but it works for some. Essentially, they bind together, they produce other products, which allows the cell cycle to continue on. We also have some cell cycle inhibitors, so these will be responsible for stopping the cell cycle if anything goes wrong. And the main 2 ones that you need to know are P53 and retinoblastoma. And these 2 inhibitors are the ones that we tend to get problems in cancer. So P53 activates other inhibitors in order to stop the cell cycle. So if P53 is inactivated, that means that your inhibitors are not produced, and therefore the cell cycle can continue despite having errors. And then jumping to RB, or retinoblastoma, once again, it's another inhibitor. So if we stop the inhibitor, then the cell cycle can continue freely, and we can run into a lot of issues. This is what I was talking about before. Such a funny word. Oogenesis or spermatogenesis. So in females, wrong one, you're going to be producing 1 oocyte and 3 polar bodies, whereas in males you'll be producing 4 sperm cells. This is the difference here. I kind of sped through this. However, there's one important point: maturation timing. So oogenesis is halted at 1 stage, and then it starts again, whereas spermatogenesis occurs rapidly and uninterrupted from the whole time. So it's just a bit of difference to do with puberty. However, both of them are produced via meiosis, and they can't survive for a long time. Okay, so moving on. We have 2 types of cell death. So we have apoptosis, or controlled or programmed cell death, as well as necrosis, or unprogrammed or unexpected death. Apoptosis is a normal process that is very necessary within our body, whereas necrosis tends to occur through trauma, chemical conditions, and mechanical failures. And there are a few differences between them. So apoptosis can result from both an intrinsic pathway and an extrinsic pathway. Intrinsic occurs through the mitochondria, whereas extrinsic occurs typically through hormones. And this will trigger the cell to start its process to die. So we have a process of apoptosis. Okay, we've got to speed up. We have a process of apoptosis, and essentially what happens is that the cell will be shrinking, the cytoplasm will be densening, will becoming more dense, and the organelles will collect together. And essentially what happens is that the cell will form these small little blebs, or these small little pockets of unnecessary material. And after this occurs, it'll break up into various apoptotic bodies. And these will be phagocytosed later on by your immune system. There are some fancy words, so can't say this, Mitosis and karyohexis, that are alternative words for chromatin condensation, as well as nuclear envelope condensation as well. So we're just comparing apoptosis and necrosis now. So apoptosis is programmed, whereas necrosis is not programmed. Apoptosis is very controlled. There's very little damage to the outside cells because no toxic chemicals are released, whereas necrosis is it's the opposite. So cells will generally just explode and release their guts everywhere. Yeah, so those are the main differences. These differences are fairly self-explanatory. However, you just have to consider the damage to surrounding cells. Okay, RJ. So now we'll be talking about Chris Wright's lecture, the unsustainable lecture, and this will be RJ, our resident international student.
Hi guys. Okay, so I'm gonna go through like fluids. I know you all didn't have a proper lecture on it, but I'll try to like summarize the main points for it. Use this thing. Okay, so now I'm just going to go through like the body fluids composition. So, you know that your total body water is about 60%, and within that 60%, 2/3 of the water is intracellular and 1/3 is extracellular. And so you need to know these 5 important ions: K plus and phosphate ion are intracellular, and sodium, Cl minus and HCO3 minus are extracellular. Just have to know that these 5 main ones are intracellular and extracellular. Okay, so I'm just going to go through diffusion. So diffusion just means a spontaneous net movement of particles down a concentration gradient, and it's affected by different factors such as membrane permeability. So the main thing that we need to focus on is membrane permeability. So in our cells, we have a semi-permeable membrane, and what it means is that it only allows certain types of particles to move through it under certain conditions. So because of this semi-permeability, there's 3 main things that you need to take note of. There's 3 different types of molecules. So there's a fully permeable molecule, such as urea, that's fully permeable. It can pass through the cell membrane all the time. And there's effectively impermeable. So what does that mean? It means that this ion can pass through the cell membrane, but is pumped back by your sodium potassium pumps. So it's effectively impermeable. Like it can pass through, but it will come back out into the blood. And finally, there's a fully impermeable ion, which doesn't pass through at all. Okay, so now going on to osmosis. So you've understood what's diffusion. Diffusion is just movement of substances down a concentration gradient. Osmosis is specifically for water. So it's the net movement of water up a solute gradient through a semi-permeable membrane. So you need to know the 3 different types of tonicity. So hypotonic just means low in solute. Hypertonic means high in solute, and isotonic means equal in solute. Okay, so osmosis of water moves from a hypotonic, which means from low in solute, to hypertonic, so which is in higher in solution, until every solution is equal in equilibrium, which is known as isotonic. Okay. So now on to osmotic pressure. Osmotic pressure is the pressure required to be applied to stop water from moving across the membrane. So water naturally moves across from a hypotonic to a hypertonic, right? So osmotic pressure prevents it from moving across. And so within your Chris Wright lectures, you probably have studied what's osmolarity. So osmolarity refers to the number of solutes per liter. So that's why you can see it's moles per liter. And there's another one which is called as osmolarity, which is moles per kg. So that's just the two different types. One is per liter and one's per kg. Okay, so now we're going to go through what's tonicity. So tonicity refers to the number of non-diffusibles. So effective osmolarity is the gradient which causes water to move. Okay, so if you have a non-diffusible, these are like your impermeables and your effective impermeables. So they basically can't pass through your cell membrane, and this creates a gradient allowing water to diffuse through from a higher water potential to a lower water potential. Okay, one thing you need to take note of is that isotonic, which is of equilibrium, does not always refer to iso-osmolar. Okay, I'll be going through this in the next few slides. So just one thing you need to take note of is, especially for medicine, is that red blood cells have osmolarity of 300 milliosmoles. Okay, so this is usually balanced, so that the blood plasma is at the same osmolarity. So your total blood water, as your total blood water increases, your tonicity decreases because your concentrations drop. Okay, so if you can look at a diagram, like when it's a hypotonic solution, your water moves out of your cells. Sorry, water moves out of your cells, and so your cells become crenated. If it's isotonic, the rate of water moving in and out is equal, so nothing happens to your red blood cells overall. And if it's a hypotonic solution, water moves into your cells from the solution, and it causes the cells to burst. Okay. So this big table is a whole summary of what you need to know for your body fluids, and I'll just be going through it in detail. So there are four main solutions that we usually use for IV. Three main solutions. I'm just adding in water so that you can understand the comparison. So pure water is hypoosmolar. Okay, so if you imagine if you have a water bath and you put a red blood cell in it, there'll be 300 milliosmoles. The concentration of the red blood cell is 300 milliosmoles, and the concentration outside in the water bath is 0 osmoles, right? So it creates a gradient, and since the water in the water bath is hypotonic, it causes the water to move into the cell, and this causes hemolysis, which is just the bursting of the cells. Okay, so now moving on to saline solution. Okay, so we usually use 0.9% saline solution for IV solutions, and it's iso-osmolar compared to the solution in your blood. Okay, and it's effectively impermeable. As you recall, like Na+ is effectively impermeable, so the particles don't move, and they begin with equal osmolarity because the Na+ cannot move into cell membranes, move past the cell membranes. So this just effectively means that when water is injected into solution, it just remains where it is, and so it creates a situation where both solutions are isotonic, both the red blood cell as well as the solution around it, which is the saline solution, is isotonic, so there's no gradient, so there's no diffusion or osmosis, and whatever happens basically is that there's an increase in extracellular fluid because the water remains where it is, and intravascular space. Okay, so now moving on to 5% dextrose solution. So this is also used in certain IV situations. So again, this is also iso-osmolar as compared to your blood. And so when you insert it, when we're given IV of 5% dextrose, initially your particle osmolarity is equal both inside your red blood cell as well as outside. Okay, but dextrose is used for respiration. So this leads to increasingly lower concentration in your plasma until the outside becomes more hypotonic. And when it becomes more hypotonic, there's a gradient now. So there's a water potential is higher in the blood compared to the cells, so water will then flow into the cells. So as you see, like initially it's isotonic, but eventually becomes hypotonic, which causes the water to flow into your red blood cells, and ultimately there's an increase in total body water. But there's no hemolysis because the process is gradual and it's spread across your entire body. Okay, so finally our 1.8% urea. Again, we always use all these IV solutions because they're all osmolar, and as you recall, urea is the only thing that we've studied which is permeable. Okay, so permeable urea undergoes diffusion with its own concentration gradient. So when you if you imagine you have a red blood cell and you put it in a solution of 0.8% urea, urea then moves down the concentration gradient, entering the red blood cell. So 50% of, imagine there's urea constantly flows into the red blood cell. The cell becomes more hypotonic compared to the solution outside, increasing the water potential gradient and causing the water to flow into the cell. So eventually the solution outside the cell is hypotonic compared to the cell and causes water to flow from the solution into the red blood cell, and this causes hemolysis. Okay, so that was just basically a whole summary of whatever you need to study. And now I'm going to give you an example using urea so that you can easily understand better. So whenever you're given a situation or given any question, just draw out this table and it'll help you better understand where the direction of flow is. So imagine we have a cell, a red cell, again, that initial osmolarity is 300. Okay, and we have blood, again, it should be isoosmolar, so that should be 300 as well. So if we add 300 milliosmoles of urea into blood, urea automatically diffuses into the red blood cell. So we technically have 600 milliosmoles in your red blood cell compared to your blood, because we assume that the blood is such a large amount, there's such an infinite amount of blood that 300 milliosmoles of urea doesn't affect blood that much, so we can take it to assume to be 300 itself. So if you notice that there's now a gradient between the osmolarity of the cell and the blood, so because there's a gradient, this creates a tonicity of 300. So now your cell is 300 compared to your blood. So what's the direction of flow of water is from hypotonic to hypotonic, so it moves from the blood to the cell. It's as simple as that. Yeah. So now we'll be moving on to David. He'll be talking about all our Med 1100 assignments.
Hey guys, welcome. I'll just quickly go through the assignments. I know this is pretty stressful all the time, given the fact that faculty is trying their best in doing online content and things like that. So I'll just go through like a rundown of what assignments you'll be getting and how to go about those. Don't stress too much. More details will be coming for those, and we'll also be putting up guides onto how best to tackle these assignments. So quickly, so this is just like a rough timeline. So in around week 5, you have the ethics assignment. So the ethics assignment, you will have 2 ethics tutorials led by the Monash Bioethics Center, and you'll learn about what bioethics are. And then you'll be needed to write around 1,000 words of an essay in response to a quite ethically ambiguous scenario, and it's worth 5% of your 1100 score. So just to emphasize before I go on further, like all the Med 1100 is pass grade only. So that just means like you need to attempt all assignments, like make sure you pass them, do it well, but you won't get a final mark on the end of your results. Like all you need to do is just give it your best shot and pass. So like relax, it's okay. These assignments should be fun and just eases you into what it means to write medically and scientifically at a university level. Probably referencing is one of the hardest parts for to do well in uni kind of level assignments. So just like make sure you figure out how to do EndNote and these other softwares that can help you with citations. We'll also put up some information on how to use EndNote as well. HKS and HKS, I'll go into detail in just one second. So just for the ethics assignment, as I said earlier, you'll just basically need to be talking about the 4 pillars of ethics: autonomy, beneficence, non-maleficence, and justice. I'm sure a lot of you already know that during your interview preparations. So you'll be given like quite an ethically challenging case, and then needed to give a point of view on the situation with account of all the stakeholders involved. Essentially, like the prompt that we got last year was this patient after surgery like needs prophylactic, so it means like preventional kind of antibiotics, and the question is he doesn't want it, but do you still give it to him? And then you have to think about like the implications involved, like the autonomy of the patient, and you have to understand these terms and use it in a real life scenario, and you will be facing a lot of these problems later on when you're an intern and a consultant. So it's often marked quite harshly, and it takes a while to get back to you, but it's just like a good exercise to start thinking and having right as a medical professional. Great. So following on, we also have the case commentary. This is probably your big whammy kind of assignment that you'll be needing. I'm not sure how this will go now or how much of an alteration it will be, as last time we went to the hospital and GP and took a patient history, but since you guys might not be allowed to do that now, I'm sure faculty will organize some other way for you to participate and finish this assignment. Because of all the virus and all the disruptions, I'm sure faculty will give you the support, so please don't worry. 3,000 words seems like a lot, but this assignment you'll only find you don't have enough words to write everything. I remember one of my friends, like they wrote like almost 10k words and had to cut it down to 3k. So don't worry about the word count, just do your best, and it's around 15% of Med 100, probably one of the biggest assignments you'll have this year. Try and enjoy it, and it'll be split into many different sections, and faculty will guide you through what you need to be actually writing within each section, including biological, so that's like the disease-based aspects of the patient's past history, social aspects like their current living situations and their social support involved, psychological aspects includes like if there's any past trauma, psychologically speaking, and how that's impacted their current day-to-day experiences, as well as how you felt during patient history, as well as what you felt of the hospital environment as a whole. So it should be quite a good experience. Just watch out for the faculty update for that. They might not even include this, might give you guys another assignment, but otherwise, just like it should be a good assignment, and just give your best. Just don't do it last minute, because this is one of those assignments. If you do it last minute, like you're gonna you have a breakdown. It's like pretty stressful if you keep it last minute. So do some every day. And finally, you also have the HKS presentation. We'll go into HKS and what this subject actually means within Med. The oral presentation is 4.5 minutes each, and in total be around 15 minutes. And everyone will be presenting this on 1 week. So this will usually be on a topic that is affecting Australia on public health scale. This doesn't mean that I think someone asked me like, oh, can we do like antibiotic resistance? Like you can, but we want to be focusing specifically on Australia and the current situation in Australia. For example, like legalization of medicinal marijuana or the aging population in Australia. And I'm sure now that coronavirus is in the media so heavily, like this will also be a large aspect that you guys can be talking about. You're being required to delegate tasks between yourselves. So as I said, with Aging Australia, we can split up into, for example, the current situation in Australia, healthcare in the future, what we can do about it, and the risks of not doing anything. So, and a person will take in charge of each one. The examiners will be external, and they will often be doctors and GPs themselves. Quite strict, but they always have questions for you after. So make sure you do this well and research very well. I don't want to go into too much detail about each of this, as we will focus on each one as it comes up, and as the current faculty academic calendars also change a little bit because of the situation, we will also adapt as the situation unfolds. Great. Now we're just having a Q&A session. So
cool. If you guys have any questions,
yeah, for like 10 minutes.
And then we'll get started on week 2 at 12 o'clock. Yeah. Also,
yeah,
I don't want to mention like med building.
Yeah. So, bit of a, I guess like unfortunate thing is that we're in the med building right now, and few people have been kicked out by like receptionist people. Like, fair enough. I don't know if we're meant to be here or not. There hasn't been like a clear like decree whether or not we're allowed to be here or not. So we're just waiting. If we get kicked out, we get kicked out, and we'll move somewhere else. And we might just like abruptly cut this and then just continue it somewhere else. I know this isn't ideal, but I think we're just trying to make the best out of this situation. And thank you for you guys for supporting us so far. And we'll give you constant updates.
But yeah, let us know if you have any questions, and we'll be here to answer them for the next 9 minutes. So that can be questions about either the content so far, or just simply about
Life.
Yeah, life, what meds like, how our holidays were, yeah, anything.
Okay, so the first question we have is from James, and he says, are those what don't during non-disjunction?
Are those?
Just curious as to what holds them together. Like, what holds a chromosome? Was that just a centromere question? Oh, okay. Oh, wait, sorry, these are old questions, they're not. Thanks, James, thanks.
Should be the 12th week.
1 second is not flattening curve. We are doing hand hygiene, it's okay.
It's okay. This is a meter and a half. Yeah, we have hand hygiene.
So, yeah, so send in the questions and we will. So we are doing hand hygiene.
I actually like need it though.
Okay.
Alright, so send in the questions. Oh, wait, quick, you know, should we do our first aid pack?
Wait, oh yeah, true. Wait, no. Okay, so David's going to introduce the first aid pack for you guys. Just one important point to make before we send that out to you guys. On there is a textbook link. Now, this textbook link is not, it is not affiliated with faculty, and it is not affiliated with MUNIS. This is a really important point to take, because as it is not affiliated with MUNIS or faculty, it's really important that we don't upload, and you're not allowed to upload any copyrighted material, or past papers or lectures, even recordings of lectures. Because all of this is under the Copyright Act. If you do upload it, there's actually academic discipline that some people have received. So when we do release this to you guys, do not. Oh yeah, well David's just released it. It's the Notion link. Just do not upload any of it onto the MUNIS drive.
There's more questions. There's actual questions. Okay, guys, I'm going to be answering some questions while they're doing that.
Yeah, just chuck it up first.
What's the easiest way to remember the bio content?
Honestly, just like learning the basic concepts, I guess. Like a lot of it, unfortunately, like a lot of med in general, unfortunately, is memorization. Like you'll find that with pharm as well, like memorizing drug names and stuff. But for bio content, I feel like just diagrams are really, really useful. Like actually just like being able to picture what's going on. And then honestly just learning the very basic concepts and just asking questions if you're unsure about anything, because you'll find that like, you know, bio like is like a advancing field. So there's going to be stuff that you don't actually need to know. So just make sure that you're always clarifying with either your tutors, your lecturers, or you can just come ask us as well. What are you doing? But yeah, so yeah, just learning the basic concepts, having a look at diagrams, watching a few videos, and that should set you up pretty well for learning bio content.
Okay, so we have a question from Vanessa. My question is, what is not an ideal solution in the body? In the body, generally everything in the body is pretty well controlled, so that means that all the fluids won't cause your other cells to explode, because that could end up pretty badly. However, that being said, water is not an ideal solution, because if you inject that into your bloodstream, you get some pretty terrible effects, as RJ was going through. Same thing with urea, which you find in your urine. Don't put that into your blood, or your brain. Don't put that anyway, just leave that in the toilet. But otherwise, your body is like fairly well controlled, so you don't need any of that.
I think someone.
Which one of you is Peter? Jeff, piss off. I am Peter. Hello.
Should we go through hand washing? Someone asked.
Do you want to actually go through hand washing?
So basically, the hand washing actual it's called a CVA. So basically, it's just pass. What?
Hand washing. Hand washing.
Yeah. Pass. Basically, it's pass fail. So you'll just be. I don't know how they're going to do this time because you're not allowed to come in a big group. So I don't know how the assessment's going to work. But what we basically got was what was it called? It was like glitter bug or something that we got sprayed onto our hands, and basically we just had to demonstrate that and you can it's basically something that you can see under UV light, and we basically just had to demonstrate that we could wash most of the glitter bug off with running water and soap. You'll get given a booklet which will basically go through all of the hand washing motions that you need to do. So it's you have to do this, and you have to do this, and you have to wash here and stuff. So basically it's just there's not much that we can really go through for hand washing because it's not actually with this. It's not actually with this. You're actually doing soap and water.
So basically, what are really concentrated solutions in the body? Stomach acid is fairly concentrated, hydrochloric acid. However, that does not affect your stomach because you do have a mucous membrane in your stomach. However, if you lose that, you end up with something called a stomach ulcer. That's pretty terrible. Other really concentrated solutions in the body. If you don't drink enough water, your urine can become pretty concentrated.
I think that's it.
Yeah.
The main one people talk about is your stomach because it's acid.
Yeah.
Don't worry much about this. This is just you basically only get saline urea. So maybe the link to application basic for now, and then just about it.
Yeah.
If we are watching lectures online later, do lecturers continue to talk during the 5? Okay, so for online lecturers, hopefully they won't because they'll know that no one's actually in the lecture theatre, and that if they actually speak over the 5 minutes, that it won't be recorded. However, with our experience, they don't really care. They kind of it really is unfortunate if that does happen. You'll just have to let faculty know, and hopefully they'll be able to do either a separate recording, and I'll be able to go through that content with you at some point. HKS additional reading. Don't do it. Don't do it.
However, have a vague idea what they're talking about because participation is marked.
It's part of 110.
It's part of meta level.
Basically,
so
the key to success for that is basically have a vague idea and be the first one to answer the tutor's questions because they won't call on you to ask further questions. So you just have to be the first one to answer.
I slept through most of my HKS. 100%, but I still passed this.
Exactly.
Do you guys have any ideas why? Clin skills further reading. Clin skills. Clin skills. Clin skills. It's
no. They set 4 chapters. Don't read it.
It's 50 or 100 words.
Don't.
100 pages. Dr. The G. Who's Dr. G? What's that mean? Oh my goodness.
It's also an additional reading altogether.
Do you have any idea why only medical students are not advised to come in?
I answered that one. I answered it up in a comment further. But I'll just say it again. But it's basically because we're as medical students we're more likely. What are you laughing at?
Mythroot. Yeah,
Mythroot's a god.
He feels like you're in a TED talk. So if you can, the lectures aren't that great for teaching, but to feel either empowered or squished, rock up to those.
He's very
charismatic.
He's very accomplished as well. He's done a lot of stuff.
Swaziland. Can you quickly go to the next thing and then we'll move on to the next?
Okay, let's go quickly to the Notion, but we've got to move on soon.
If you've got more questions, it's all good. We can
We're
ask
gonna
them
answer
later.
It's just it's because we're comparing against blood.
Okay, so here is the first aid kit. So a bit of a blurb. We have some useful links for you guys, so some useful resources. If you click into the box, it will pop up like this, and the link will be within the box, and a bit of a description from David and the rest of us. Once again, the textbook. Here's the warning. Once again, please do not. We will throw you under the bus.
We will. Like, that's not a joke.
Yeah, that's it, not a joke.
Please.
Here's some advice for you guys. David's a bit... Yeah, David's not actually that. David's not actually dumb. He says he is. But yeah, this is the document. So hopefully that may help you guys get through some things in the coming few weeks. But in the meantime, we should probably get started on week 2. And we'll be starting that off with Central Dogma from yours truly. Hopefully faculty doesn't kick us out, because I know you guys really want this stuff. Alright, so once again, you guys can follow along with us. David is nervously checking through the blinds for people. Okay, let me just get set up here. Okay, so let's get started. Apparently there's someone coming, but we'll get through as much as we can as possible. Okay, so central dogma. Okay, useless. Okay, so DNA structure is made of 3 parts: phosphate, sugar, and nitrogen base. The important part to note is that phosphate is your 5', and the sugar down here at the 3rd carbon is your 3'. That'll be important for directions. Nitrogenous bases, you have A, T, C, and G. A and T go together, C and G go with each other. They are also in uniform diameter, and they're in an anti-parallel arrangement. So that means it's like a ladder, except both directions are heading in opposite directions. So an easy way to kind of think of it, it's like a really straight freeway. Some cars are going in one direction, other cars are going in other direction, but it's both parallel to each other, so they don't hit each other. 5 and 3 will join together. And this is what it looks like. This is how your DNA condenses, so it wraps around some histone proteins and into your chromosomes. DNA has some essential functions. So storing genetic material is one of those functions. Gene expression, it has to be able to be replicated, and it also has to be susceptible to mutation. The reason it's important to be susceptible to mutation, despite the kind of negative connotations around the word mutation, is because it helps to introduce some genetic variety at times. And structural features is what we've been going through. It's important to note that the backbone between the 3' and the 5' are all covalent bonding, whereas the forces between nitrogenous bases are hydrogen bonds. So hydrogen bonds are weaker, and that will allow for a few things later down the track. If we compare that all to RNA structure, however, RNA is slightly different. RNA is single-stranded, and it has a ribose sugar instead of deoxyribose. The uracil base also replaces T, so U will replace T, so there's no thymine at all in RNA. And we have 3 different types: mRNA, tRNA, and ribosomal RNA, or rRNA. Okay, so we will be first starting off with DNA replication. And this is slightly different between prokaryotes and eukaryotes. So in prokaryotes, as you know, the chromosomes are circular. It's a single chromosome. So therefore it has one point of origin. So it starts at one point and it circles the whole way around. Whereas in eukaryotic chromosomes, there are multiple points. The reason for this is because there's a lot more genetic information, as well as there are multiple chromosomes. So you have to start at different points to be able to get through everything effectively. It also serves an evolutionary purpose because it avoids the loss of a complete origin site. Because if there is a mutation at the origin site, you lose it, and if you can't replicate, then you essentially die. So we'll start off with the DNA replication process. It's a bit complex, but we do have another we have a diagram on the next few slides, which will help us to get through. So if you imagine 2 pieces of DNA, one strand of DNA connected with another strand, we have a template strand as well as a coding strand. And what happens is that an enzyme called DNA helicase will attach to these 2 strands. And what will happen is that it'll unwind the DNA into two separate strands. So it breaks the hydrogen bonds that we previously talked about. There are also some smaller proteins that attach to these individual strands, and they prevent it from rejoining together. Following this, we have another enzyme called RNA primase, and it attaches to the template strand of the DNA. And it will catalyze short, oh, sorry, that was a mistake there. RNA primase will attach to both strands, both leading and lagging strands, and I'll explain what those terms mean. And they'll essentially create short RNA primers to kind of provide a starting block for the DNA polymerase, which we'll talk about soon. These RNA primers are later broken down and replaced by DNA. In addition to this, following the RNA primers, so if we recap, we have two separate strands of DNA, and we have RNA primers on each strand. Now, following this, we have DNA polymerase, another enzyme, attaching to these primers, as well as a sliding clamp. A sliding clamp is essentially just it's just like a holding protein that'll keep it onto the DNA and stops it from falling off. So DNA polymerase will always run in a 3' to 5' direction, and it joins it in a 5' to 3' direction because it has to be anti-parallel. And because it always runs in a 3' to 5' direction, it'll be an opposite direction on each leading and lagging strands. And we'll have a diagram because it's very difficult to explain through words. But I'll try to explain it now. So as I was saying, DNA runs in opposite directions. So therefore, at one end, there'll be 3' and 5' on the opposite strands, and at the other side, there'll be 5' and 3' on the opposite strands, because it has to run in opposite directions. So because these two strands run in opposite directions, and DNA polymerase only runs in 3' to 5', that means that DNA polymerase will run in one direction on the top strand, and then in an opposite direction on the bottom strand. On the lagging strand, there's a bit of jumping, because as it moves towards the 5' end, it has to jump back to bits that were previously unwound and copy that. So it'll essentially move like this, and it'll jump back, move, jump back, move. Yep, and I'll show you a diagram of that. Okay, so this is what it looks like. So these grey strands here are your original DNA strands. So as you can see, it was 3' here, and 5' here, and 3' here, and 5' up here. These light grey structures here are DNA polymerase, and as a reminder, DNA polymerase only runs in 3' to 5'. So on our lagging strand at the bottom, it'll be moving like so towards this direction. And as it moves in this direction, it has to jump back closer to this junction point here. And the reason for that is because our overall direction of replication is heading towards the right. So our DNA polymerase has to start here at this primer, and it works it all the way to the end. And then because it's at the end, there's nowhere else for it to go, so it has to jump all the way back to newly exposed DNA. Whereas on the leading strand, it's a lot simpler. It simply joins at the 3' side, and it just runs along as the DNA is unwound by the helicase. However, at the end of these lagging strands, there's going to be a small bit of overhang. And the reason for that is because to synthesize this section here, the DNA polymerase would have to bind over here, where our 5' currently is. However, there's no place to attach our RNA primers here, so therefore we actually miss out a bit of DNA at the end of our lagging strands at the 5' end. However, we have an enzyme called telomerase, which will essentially synthesize an extra bit on our original strand up here, where the 3' bit is, and then our RNA primase will come in, create a primer, and that will allow our DNA polymerase to synthesize this last part so that we are able to copy all of our DNA. Okay, so a bit more information about DNA polymerase. There's a question. Do you want to answer? Does lagging? Lagging, if we consider from direction from left to right, it'll be 5' to 3'. Yeah, that's the question. Your lagging strand runs 5' to 3'. On both sides, okay. So the reason we have a leading and a lagging strand is because our original DNA sections here, the bottommost strand and the topmost strand here, run in opposite directions. So because they run in opposite directions, but our overall direction of replication is going to the right, that means our DNA polymerase must move in opposite directions on each strand. And because of that, you result in these shorter fragments compared to a longer fragment here. I'll find a video following this and I'll post it up for some additional resources to make it a bit more clear. And perhaps if we have time at the end, we can go through it again, but we have to keep moving, unfortunately, and onto the role of DNA polymerase. So this is the enzyme that copies the DNA, and it's like a hand shape. So it's like a hand gripping a handrail and it moves along. So the finger regions will read the nucleotide bases so that the correct bases would be made, can be attached to that. And the energy for this is through deoxyribonucleoside triphosphates. And essentially what happens is that your two phosphates will be cleaved off, and that'll provide energy throughout the thing. So you can see that here, as we're making the strand, we have this triphosphate nucleotide, and it joins on through a condensation reaction. Wait, is it a condensation reaction? Yes, it is a condensation reaction. And we break off these two phosphates, pyrophosphate, and that provides energy. So following this whole process, we have to check our DNA so that we don't end up with any errors. So we have three types. We have proofreading, mismatch repair, and excision repair, and it all essentially depends on the timing when it occurs. So proofreading will occur during replication, whereas mismatch repair will occur after the entire replication has occurred. And lastly, excision repair occurs throughout the cell life to fix any damage that occurs through chemicals or radiation or otherwise. Okay, so we kind of just ripped through DNA replication. And now we'll be talking about the actual central dogma, which I believe you guys didn't quite get up to in this morning's lectures. So, take a shot every time it says, okay. Thanks, guys. So, we'll be talking about transcription and translation. So, in the first stage, transcription, it's a fairly short process. And, oh my goodness, I hate this so much. So, essentially what happens is that we'll be dealing with the template strand. Template strand at the promoter sequence, and the RNA polymerase will recognize this sequence and it'll bind to it. And this process is affected by transcription factors. So this enzyme, RNA polymerase, moves in a 3' direction to a 5' direction. And it synthesizes in the opposite direction. Once again, as a reminder, RNA has uracil rather than thymine compared to DNA. So this process continues on through RNA polymerase, and it stops at the termination sequence, which involves a protein complex. It's essentially just a protein complex that will stop the process. As a result of transcription, we can either produce messenger, ribosomal, or transfer RNA. But the main one we'll consider is messenger RNA. So a bit more information about RNA polymerase itself. Prokaryotes only have one type of RNA polymerase, and that does everything, whereas eukaryotes have 3 different types that do the 3 different types of RNA. An issue with RNA polymerase overall, though, is that it does not proofread, so you can end up with a lot of mistakes here. So following transcription, we have some modification. This was covered in Bio 3.4. This whole process is covered in Bio 3.4, but for those who do not know it, it's quite confusing. So at the end of it, we end up with a single strand of RNA, so mRNA, and we have a 5' end and a 3' end. So a methyl G-cap is attached at the 5' end, and a poly-A-tal is attached at the 3' end, and that's essentially just a sequence of adenines over and over and over again. So the purpose of these two caps is to prevent degradation and damage as it leaves the nucleus because of the various chemical reactions that occur there. So yeah, that's the role of those two caps. We also have SNRPs or SNRMPs that form spliceosomes to cut the introns out of the mRNA strand and join the exons together. The reason this is important is because introns are non-coding regions and therefore not used during translation to produce proteins, whereas the exons are. However, that being said, exons can be joined in various patterns to provide various different proteins. So as an overall outline of translation, which is quite a large process, it has four main stages. So activation of tRNA, initiation, elongation, and termination. A question, James. Are SNRPs proteins or just another generic molecule? They are proteins. Okay, so essentially, if we only have one mRNA strand, to increase the efficiency of the whole process, we can have more than one ribosome moving along that one strand at a time. So therefore, we can produce multiple proteins from the one, mRNA strand. But first, we'll talk about the various types of RNA. So we have transfer RNA, and it's a single-stranded tRNA that is bound to itself, and it connects amino acids, mRNA, and the ribosome together, and this allows for the process of transcription. It contains an anticodon sequence, which is complementary to the codon sequence on the mRNA. So this will allow for specific recognition of these sites, which will code for a certain amino acid, which is attached up here. So these you can view as kind of trucks to deliver amino acids to the ribosome. Now, talking about the ribosome itself, it's actually made of two different parts, a smaller subunit and a larger subunit. These numbers here about how much RNA and how many proteins they include aren't too important. However, these three sites, which you can remember through APE, are very important. So the first site, amino acyl tRNA, is where the tRNA containing the amino acid will bind to the mRNA codon, as we saw in this previous slide. So this would be the APE slide, the amino acyl tRNA. Then it moves on to the second position called the peptidyl tRNA, and this is where the amino acid is joined onto a current polypeptide chain. And then it moves on to the next stage of exit, and the tRNA is removed, and it can go to get another amino acid. So you can see that here we have our larger subunit and our smaller subunit and our three sites, A, P, E. So the first stage of translation is activation of tRNA. And this is essentially where the tRNA will bind to a specific amino acid. So we have these enzymes, amino acyl tRNA synthetases, binding the amino acid to the tRNA. And these are very specific. So one tRNA can only hold one amino acid of a specific type. That's a diagram that you don't really need to know, but it helps illustrate the picture. The second stage is initiation, and we use an energy form called GTP, or guanine triphosphate. It's essentially the same as ATP. And what happens is that we have an initiation complex made of the first tRNA, so always your start codon methionine, your amino acid of methionine, the mRNA strand, and a small ribosomal unit. And what happens is that it'll bind to a complex or a sequence called Shine-Dalgarno sequence. And the start codon will always designate methionine. And following that process, the large subunit will join on. So the main point to take from that is that the small subunit will join on first, and then the large subunit will join on later once the first amino acid is found. So you can see that here we have the initiated complex binding with methionine, and then the large subunit will join at a later time. And following this, the ribosome will continue to move down the mRNA strand. So as we move along, the ribosome will read the specific codon on the mRNA. A specific tRNA will come on with its complementary anticodon and its specific amino acid. And the two amino acids will be bound together through a peptide bond, or covalent, through a condensation reaction. And then following this, the tRNA is released from the each site. And after we've moved through the whole mRNA strand, the ribosome will reach a stop codon. And these stop codons, UAA, UAG, and UGA, will not code for any amino acid. And because it doesn't code for any amino acid, it allows a release factor to bind on, and that results in the ribosome disassembling between a small and large subunit and the polypeptide being released. And following this whole process, we now only have a single strand of polypeptide. So this, once again, will undergo some modification afterwards. These words aren't too important, but they're good to mention as they come up in one of the active learning sessions that I'm not even sure if you'll have anymore. But it'll be formed into what we call a secondary, tertiary, or quaternary structure. So secondary structures are beta pleated and alpha helices, tertiary is 3D, and quaternary is where you have multiple polypeptides attached together. If these polypeptides do require special conditions to fold, we have chaperone proteins, which are kind of like, I like to think of them as like mini phone booths, where they can go in, it's like a closed box, and it can provide the correct conditions for folding. Proteolysis, glycosylation, and phosphorylation are just adding various groups onto these proteins. These are mostly where I got my pictures from because, yeah, that's useful. But we'll be doing med law. I'm sure there are questions for central dogma, but I'll be answering these afterwards, later on. Yeah, but Jating's answering most of them. So medical law will be taken by RJ.
Yeah, hi guys. I'm going to go through med law since Jating's not here. So for med law, there's a few things that you need to take note of. So firstly, consent. So all health practitioners and us as medical students, we are legally obliged to obtain a valid informed consent before any medical treatment and procedures can be provided. So what's the definition of an informed consent? It refers to an individual's agreement to a procedure and treatment, and this is after they have full disclosure of all the risks, benefits, alternatives, and consequences of the outcome. So there are three main elements of a valid consent. Firstly, voluntary. So the consent must be given freely, and the patient cannot be under any influence of any drugs, alcohol, or stress, and it cannot be forced. Secondly, it must be informed. So as I was saying, like, the patient must know all the risks, all the benefits, alternatives, consequences, everything that's pertaining to the treatment. And finally, capacity. So the patient must be competent to make this decision. So legally incompetent patients would include patients suffering from mental illness or intellectual disability, unconscious patients, or if they're affected by substance or drug abuse, or minors. Minors is those under 18. So there's some exceptions to this particular law. So a child under the age of 18 can be assessed as a mature minor according to common law, allowing the practitioner to act in the minor's best interest. But this law depends on how old the child is. So closer to the age, then the child can be exempted from this. Okay, so there's types of consent. So there's the implied consent. So it's not explicitly given, so it can be gathered from the patient's words and actions. This is most common, especially when you go and see a GP. If you want to take their blood pressure and they put out their arm for you, that's sort of an implied consent. Oral consent is when you clearly ask for consent and the patient clearly agrees to it, and this is often gained when there's doubt of implied consent or if the procedure is more invasive, such as the chest X-ray. And finally, there's written consent, and this is particularly for treatments which are invasive, such as surgery. Okay, so when can consent be waived? So in emergency circumstances, to avoid serious and imminent harm to patients by giving CPR, or when it's a necessity. So when the situation is not necessarily a threat to life, but no action may result in a serious impact. So the doctor must be able to argue that any reasonable person would have taken action in the situation. But for certain groups of people, legal consent can be obtained from someone other than the patient themselves. So for minors under the age of 18, there's usually a legal guardian who's consulted, or if the patient does not have the mental capacity to consent and is not in an emergency situation whereby the patient's life is in danger. So if you fail to obtain consent, there are legal consequences. So there are two legal consequences. Firstly, battery, which is the intentional contact with another person's body without consent. And a patient who consented to unilateral, for example, if a patient consented to unilateral mastectomy receives bilateral mastectomy, that a patient can then sue the doctor in court due to that. And another case is assault, where a patient has a fear of imminent physical harm or misconduct. So if a doctor threatens a Jehovah's Witness to receive blood transfusion, the patient now feels the doctor will perform the blood transfusion without the consent, and then they can again go to court. So the consent can also be deemed inadequate, and the doctor can be charged with negligence because the doctor fails to meet the expected duty of care. So issues with consent: if there's refusal of treatment, if the patient satisfies all elements of consent but chooses to refuse treatment, then the right of refusal must be observed, given that the patient has the right to self-determine. And patients who do not wish to be informed, a patient can wish not to know the details regarding the treatment when they consent to it, and but in this case, the health practitioner should insist on providing the basic information, and if the patient still persists on being left uninformed, the doctor should then record this incident in the patient's medical records. So doctors are required to hold doctor-patient confidentiality, and this is critical in maintaining a doctor-patient relationship. So there are situations when doctors can disclose information, and there are situations where they must disclose information. So situations where they may disclose is when the patient has consented, patient's details are de-identified in interest of public health, and you can just read on for that. But like whatever you need to know is the legally obliged to disclose. So when there's notifiable diseases such as HIV, like currently the coronavirus, medical malpractice, and reportable deaths. So reportable deaths are when the doctor has to legally report to the coroner. So such cases are when there's unexpected or unnatural death, or if the death occurs during the medical procedure, or if the patient is placed under custody of the Department of Justice immediately before death, or if it's an unknown identity. So there's a few sources of law. So there's common law, which are rulings which are based off preceding cases involving similar legal principles, and there's a hierarchy. So lower courts are bound by the decisions of high courts, and there's statute law, which is legislation passed through Parliament. Okay, so now moving on to the medical interview, which is
Hi guys, I'm just gonna quickly run through the medical interview. You're gonna go through this in a lot more detail in your tutorials. You'll be basically breaking down the whole medical interview across 3 or 4 shoots. So don't worry if this is all a bit unfamiliar. You have plenty of time to actually learn all of this before your CSSC at the end of semester. So no stress. Okay, so basically the first part of the medical interview, this is what you're going to be basically learning this week. So the first part is a preparation phase, basically collecting your equipment, basically stethoscope usually, and then performing hand hygiene, which is, we'll upload a video on that. Establishing initial rapport, that's basically just introducing yourself, making sure you have the right patient, because it's kind of bad if you don't ID the patient beforehand. And we'll basically be going through how to introduce yourself, and then the last step is identifying why the patient has actually come in for an interview. Okay, so basically this is what you need, this is how to introduce yourself. This is kind of, you need to just memorize this, but it'll basically become second nature, because all of us here can basically recite this within 30 seconds and in in 2 breaths. We can just say this whole thing. So you can like read that for yourself, but basically some key things is the first sentence, which is introducing yourself. You need to say your first and last name. Some tutors won't tell you that, but like my tutor did, and apparently it's a thing, so just say your full name. And say you're a second year medical student from Monash University, and you have to tell them what you're doing today. So basically what you're doing is just doing a brief medical history, and this is the part where it's informed consent. So you need to actually say what you're going to be asking them. So that's just a brief summary. So like what's brought them in, past medical history, social history, and just ask them if they're okay with that. And then you have to ensure, like you tell them that it's going to be confidential because we've just gone through med law and all that stuff. So everything has to remain confidential. And then you need to get some identifying information. So these are the 4 identifying like data points that you need to get. So you need to get their full name and what like their preferred name is. You need to get their age and their date of birth. You need their current occupation or their previous occupation. And you need to identify whether they are of Aboriginal or Torres Strait Island origin. This is the exact question that you ask, because there's certain ways people phrase it that like don't come across as very like respectful. So basically just like ask this, and then you also need to identify whether they are of Aboriginal, Torres Strait Islander origin, or are they of both. This is, so HOPC basically stands for History of Presenting Complaint. That's basically asking them why they're coming in today. So you usually just ask, okay, so like what's brought you in to come see the GP? And you'll learn this acronym, WWQQAAB plus ICE. And basically these are all the questions that you'll ask to get a really kind of detailed overview of what's actually going on. So I won't go like into that in detail. You'll be going through this in a lot more detail in your tutorials, and you'll actually be getting a lot of practice doing role plays with each other and in class. I'm not sure how that's going to work, but like, you'll get a lot of practice and a lot of different scenarios. Okay, so this is just a little quick overview about hand washing and the injecting CBA. So like I said before, this is a pass or fail. So all you have to do is just don't make any major mistakes and you'll pass and everything will be fine. So hand washing, you just need to know the 5 moments of hand hygiene, which is like, you'll get like a sheet, but it's like before you greet the patient, after you greet the patient, before you like start a procedure, after you do a procedure, and like after the patient leaves, basically. You'll have like fancy like hand washing maneuvers. It'll all be given to you in like a tutorial document. Injections, I don't know how they're going to work, but please, when you actually start doing them, please don't get a needle stick injury because it's an immediate fail and you'll have to do it again, and that kind of sucks. We'll be uploading a video that'll go through basically how to do injections, so don't worry about that. Okay, I will move on, and now we're doing fields. Oh, really? Oh god. Alrighty, amazing. Okay, so cellular, energetics, and enzymes they haven't covered it yet. Have you covered this yet? Okay, so huh? 20 seconds. Okay, anyway, so I'm actually going to skip over a lot of this stuff, especially the enzymes things, because if you did VC chemistry, it was VC chemistry, yeah. Yes, if you did VC chemistry, you should already know 99% of this. So, yeah, once we upload this slides, have a look through this yourself. Not particularly high yield. Only point on this slide is that for G, think about delta H, and it's basically the exact same thing. And anabolism and catabolism, know both of them. So catabolism is breakdown, anabolism is, wait, did I get this the wrong way around? No, I did. No, that's fine. Anabolism is breakdown of synthesis, and catabolism is breakdown. Wonderful. ATP is basically your energy currency, as I'm sure you already know. Lots of words for a very simple concept. We'll skip over that as well. Rates of reaction. you did BC chemistry, you already know this. So you have your concentration of substrates until it becomes, you know, saturated and then rate plateaus, and enzymes and a bunch of different factors affect the activity of enzymes. And we will be going through that. Enzymes, once again, BC chemistry, decrease the activation energy barrier, so a greater proportion of collisions are successful, blah, 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 blah. They're specific, know that point, and you also need to know how they work. So they orientate your two substrates, they can add physical strain or add electric charges. Factors affecting the activity of an enzyme, BC chemistry, so you have the concentration of substrates, the allosteric modulation, so actually memorize those few things, so you can have the reversible, non-covalent binding of a modulator at a site other than the active site, and it can be, what's it called? It can inhibit the enzyme, or it can activate. What's the better word for activate? Well, yes, it will activate the enzyme. Blah blah blah. Terms of enzyme inhibition, know about prosthetic groups, permanent inorganic factors, and coenzymes. Yeah, definitions, just remember them. Inhibition, yeah, enzymes are very important, know about them. They can be reversible, irreversible, memorize all of that. They can actually, there have been exam questions on it, so actually memorize the definition. It's not very hard anyway, you'll be fine. Enzyme inhibition, lovely little diagram to summarize it, have a look through it. Okay, fuels, fuels caused a lot of problems last year. People felt very, very confused the entire time. It was actually kind of poorly taught, but the main point is, it's basically all about metabolism. It's just about how we take in food, or how like energy stores within our body, so like all the beautiful fat we have on our face and our stomach is actually used, and how we can actually, you know, function in our day-to-day lives. Okay, so yeah. So we will be going through this step by step, very slowly, but this diagram basically shows you everything you'll need to know by the end of how many weeks, whatever. Yeah, at the end of 2 weeks, but it will make perfect sense. Very, very straightforward, but that all puts it together. Okay. Yes, it will be fine. Metabolism is the combination of chemical reactions occurring in our body at any given time, blah, blah, blah. 3 main fuel sources. So you will have to know about each of them. So carbohydrates, how they use, what are the different processes that we use to break them down and create ATP, lipids, and proteins. So there are 3 different pathways, and they are connected at different points. But go through them one by one and it will make perfect sense. So it says some of these processes are for carbohydrates, lipids, and proteins. So they are separate processes. Don't mix them together and it'll make more sense. Require carrier molecules, so NADH, FAD, and ADP, blah, 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 blah. And they transfer electrons between reactions. So don't worry about them too much. Memorize them. A lot of this is just memorization. So glucose metabolism, where does it come from? We eat glucose. I eat too much, and mobilization of stored glycogen. So, you know, when you're going for your run and you have like all the glycogen in your muscles, carb loading, yep. Steps, glycolysis is first, then your Krebs cycle / citric acid cycle, same thing, and oxidative phosphorylation. So your ETC and your chemoosmosis, fancy words for the same thing. Glycolysis, okay. Wait, oh, my favorite little slide didn't come up. Okay, never mind. Okay, so when Nirma goes through her slides, she will have this yellow box that says, not essential, no key steps only, but she doesn't tell you which steps are essential. So what I've actually done is there are a few like bolded things. So yes, in general, you don't need to know details, but then there are some tiny little details that might actually come up on your exams. So for glycolysis, it's a series 10 enzyme catalyzed reactions. Don't need to know each of them. Oh shoot, oh never mind. It can operate in the presence or absence of oxygen, so know that. So it converts 1 carbon molecule into 2 pyruvates, so no inputs and outputs. I should probably update those slides, but whatever. First 5 steps are called the preparatory phase. The last 5 are the payout phase. So know those 2 words, preparatory and payout phase. And also know these random numbers. So steps 1 and 3 use up 1 ATP molecule each, and steps 7 and 10 produce 2 ATP molecules each. Don't need to know why or how, just know these numbers. Wonderful. Fate of pyruvate. Oh yes. So once you've gone from glucose, change into 2-pyruvate, 2 things can happen to that pyruvate. So if you have enough oxygen, I'm sure you already covered this in bio, enough oxygen, it goes through the intermediate stage and it's converted into acetyl-CoA. Without, it undergoes anaerobic respiration. So anaerobic respiration in animals turns into lactic acid. You're going for a really fast run, ouch, your legs hurt. Ideally, yes, we like, so if it has enough oxygen, it becomes aerobic respiration, which is good because it produces lots of ATP. Anaerobic respiration is not very efficient, so we don't like it very much, and lactic acid hurts. There's another diagram, have a look through it later. It doesn't really explain much other than the fact that, oh yes, another key point is that the conversion from pyruvate to lactate requires the enzyme lactate dehydrogenase. Memorize that. Okay, Krebs cycle, citric acid cycle. Once again, you'll get that yellow box that says need not to know key details. Well, which key details do you need to know? You need to know the inputs and the outputs, and also know the first step and the last step. So citrate is the first thing after acetyl-CoA, and oxaloacetate is the last. Memorize those. You don't need to know the other steps. Other things to know: each citric acid cycle produces 3 NADH, 1 FADH2, 1 GTP, and 2 CO2. Memorize inputs and outputs. Keep in mind, you also need NAD plus for the citric acid cycle to work. That will become more important later on when we get into like alcohol and stuff like that. And once again, the cool thing about fuels is that you'll actually get to learn about, like, why do Asian people get Asian flush and how does that kind of stuff work. Okay, oxidation of pyruvate. So this is when we have enough oxygen. So we've gone from glucose, we've gone down to 2-pyruvate, and we have enough oxygen. So now we're going to go through the intermediate step. So going from pyruvate to acetyl-CoA. I've put the inputs and outputs there, memorize those, and the key enzyme is pyruvate. Oh, did I get that wrong? Anyway, I'll fix that up if that's wrong. Pyruvate dehydrogenase, and key detail about this is that this enzyme requires thiamine, a group V vitamin. And exam, they might be like, wow, what happens if you don't have enough group V vitamin, thiamine? And you can be like, oh, well, you can't go through the oxidation of pyruvate step. Wow, that's so bad. Oh, wow, what the patient's going to feel like. Okay, no, that's done. Okay, so what happens if we don't have enough oxygen? We'll go through the lactic acid cycle, the Corey cycle, so fancy words. Fermentation, this occurs in the cytoplasm. Yes, memorize that fact, it could show up on your exam. Once again, inputs and outputs are there, memorize them. Oh, I did definitely get that wrong because the enzymes aren't the same. I'll fix it up, don't worry about that. Yes, memorize the inputs, outputs, enzymes, and where it occurs. So basically all the points there. Not very, very high yield. Actually, yeah, it is actually kind of high yield. Yes, it is high yield, remember that. Conversion of pyruvate to lactase requires low yield. Okay, next part is electron transport chain. So you don't actually need to know too much about this. You can memorize the names of the different proteins if you want. I personally didn't, and I still did fine. Yeah, just read through it. Not very high yield. No, I'm teaching them the high yield content. And then you also have the ATP synthase, which is a key enzyme involved in the ETC, blah, 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 blah. ETC steps, you can read through this if you want. I'm not going to read through it. It's just a series of steps. I've highlighted some things, so those are the key points. You can memorize them if you want. It's up to you. I didn't. Actually, yeah. So this stuff, you might want to go through it because you might have an active learning. So we had an active learning last year where nobody had any idea what was going on, and that basically covered this. Yeah. But yeah, whatever. ATP synthase. Just read through the notes. You've quite low yield. Okay, wonderful, that's done.
Okay, hello. Okay, I'm back. So just for HKS intro, we'll quickly go through it. I know you guys haven't done it, but you guys will have the amazing Mike 3 as he will slow down his voice in a very dramatic fashion. Okay, so just to begin, like, it's really important, you'll hear this statement so many times, like, what is health, right? So according to the World Health Organization, health is a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. So that's really important. Like, it says a complete absence of physical, mental, and social disease. So it's like full well-being, and obviously that's like really hard to achieve, right? So this is a limitation of it, and it's like, what does it mean to be well? Like when you say like you're mentally well, like is it having stress from like school, like or like uni? Is that still considered being well? Like how do we measure it? It's a bit qualitative, so we can't measure it quantitatively. And it's the same thing with like UMAT logic. Like if you see complete or like all, like it usually has flaws since nothing is like ever 100%. Yeah, but the strength of the good thing about this definition is that health, it considers all aspects of health, right? Rather than limiting it to a physical sense. Like if you break your bones, like you're not healthy, but if you've got a mental health problem, like that's also not healthy. So it's considering all aspects. Okay. Yeah, so measurements of health, what can we measure health by? So firstly, life expectancy. You guys can read the definitions, I just quickly explain it. So in the blue, it's just like a way to remember. It's like, I want my refund back if I don't get X number of years on this earth. Like it's on advertisement. So the advertisement being the life expectancy rate at birth. It's like what you're predicted to get, but it's not always accurate. Disability adjusted life years means like don't dilly-dally. So like you're wasting your time, right? Like this disability will be taking away your time. Otherwise you will lose healthy days or die too early. So one dally can be just thought of one lost year of a healthy life. Healthy life means like you have normal day-to-day abilities such as cooking, buying groceries, like doing exercise, meeting out with people. You need to know what each of these means, but faculty hasn't tested students to calculate dallies by any means. Years of life lost, you guys can just read through this. It's just like how many more lives you should have lived by, and just read the years lost to disability. Don't memorize formulas, just know what they mean, and apply to a lot of medicine, especially in a public health sense, given the fact that coronavirus is like in the media a lot. So just like know what they mean. You might not need to memorize it completely. Alright, this is pretty important. So incidence and prevalence, like what do they mean? So incidence basically means it's like how many new cases of certain diseases appear over a given period of time. So if 50 children were diagnosed with autism in the community in 2019, so those are 50, right? That's the incidence, the new cases. They were recently diagnosed. Compared to prevalence, which is like in this community in 2019, there has been 500 children with autism. So like that includes people from before as well as the new cases within 2019. If you look at the bathtub, it's like a pretty good way to think about it. We'll go into more detail, especially in semester 2 when this becomes a big thing in population health. And if you are not sure about the examples and things like that, I've given a bit of calculations just to guide you through the mathematical aspects of that, but that is not necessary. Health inequity and equity, you guys will definitely cover this in HKS. I know the camera thing is covering the photo, but you guys can look at it later. So equity basically means everyone gets what they need to fulfill their full health potential. This means like if you're living in a third world developing country, developing like health-wise country, you will need to have more support to get the same health outcome compared to anything else. Yeah, so the photo is there now. Whereas equality is like everyone just gets the same, but obviously that doesn't make sense, right? Like if someone living in a rich suburb, like their access to GP is much, it's easier to get, whereas those in developing countries will have a difficult experience. So the example, like you guys can just have a read, it's just about like having a good distribution of resources as well. Global forces, Mike 3 covers this in the first lecture. So firstly, geopolitics. This just includes like conflict, displacement. Just think of the big events happening. This also includes like even like coronavirus now, like people talking about how that's just dislocating people from their homes. Social cultural factors is like human rights neglect. If human rights doesn't exist, like poor health is inevitable, and human health determinants, it's like social determinants. Like this includes like sex, age, race, sexual orientation, cultural background, religion. So any aspects that you are kind of born into that will affect your health in a negative or a positive way. Environmental change, this includes climate change, as well as so climate change includes rising sea levels, glaciers melting, bushfires even. All of these are environmental changes, and they will drastically impact us. Look at bushfires, for example, people getting dislocated, dislodged from their current living environment. And historical influences, colonization, and the Aboriginal people. You guys will be going to that in week 8, I believe. Is that it? No. And lastly, one of the last slides is medical epistemology. Sounds like big words. My 3 loves adding these, and you need to know how to actually use these in your own sentences and discussion. So the nature epistemology just means the nature and origin and the limits of knowledge. It's just the way we think about things and what is knowledge and what we think about it. So this is definitely content for the big brain meme page. So medical epistemology, thought structures within medicine, like paternalism versus patient-centered care and scientific methods and things like that. Whereas ontology is the study of being, what exists. So think about how something exists and how it fits into the wider frame of things. This is really important, how one thing is only a cog in this entire mechanical system. And see ontological multiplicity. This is one of those big words, but it can be broken down quite easily. I won't cover the Habermas's thing, it's pretty low yield, but just have a read. Yeah, and we'll cover it next week as well, once again in HKS. The epistemic traps, this is just the ways that you think in which there is a fallacy within it. So positivism means that only things I experience are real. So you can't have there's only logical proof for it. So when you're looking at something and you think, okay, if I only see it, then it's real, but we don't take into account individual experiences. And the main issue is that it claims that scientific fact is 100% accurate and certain, but ultimately there will always be things that are not fitting nicely into the usually reliable scientific framework. And human experiences and things like that is going to be a flaw of positivism because you can't actually see it, right? And inductive fallacy is just overgeneralization. You see a group of black swans and you're casting your view that all swans are black. Obviously that is, you're inducing, you see swans and they're black, so therefore they're all black. Obviously that's not right. Monogenesis, Mithra will cover that in more detail. He didn't really explain it that well in our one, so I'm assuming that's very low yield as well. Okay, and yeah, just don't worry too much about these things. Just know what they mean and be able to use them in a discussion or essay in the part of your exam. You will probably be needed to write around 200-300 words for HKS as a short essay within your task. Or how do I?
Okay, questions.
Oh yeah, questions.
So
Right, we're done.
last question and Q&A before everyone except me has to go to anatomy tute.
No, we do.
We have to use our computer. Yeah.
Does anyone have any questions just before we finish up?
Okay, in the meantime, what I've done is on our Facebook group description, there's both our Google Drive link, but I've also added a calendar link. So that should give you a rough schedule about what topics we'll be covering. So it'll list the location once we figure out what to do with that, but it will also list the covered topics during that week. So check that calendar for updates in terms of events. However, you will need to use your Monash email to access that calendar, and you can probably add that to your own personal calendar. So you can keep up to date with all that stuff.
Any questions before?
Next week, we're still trying to figure out the best way to do this at a permanent location where we can stream. This place is really their building's really good for streaming, but once again, it's I'm surprised I haven't been kicked out yet. So yeah, we're glad to have made it through this one, and thank you guys so much for tuning in to our PSP today and supporting us. So yeah, thanks a lot.
Yeah. Any questions?
Yeah. Any questions?
Any questions? Doesn't have to be about academic content. In the last 6 minutes, I really have to pee. Okay, Zoe needs to go pee.
Yeah.
If we get kicked, we get kicked.
Okay, we've already finished.
The door's locked.
Is it locked?
I locked the door.
Thanks, James, for making that centralized thing. It's pretty good. May24.xyz/PSP.
Yeah, same for James.
Any questions about our life, about our holidays, about
if you do think of any other questions, throughout the week, feel free to message us on Facebook. All of us are like the admins, so you can just find us there. Otherwise,
we'll put the upload a Google Forms.
Yep, there's a Google Forms within our PSP Google Drive. So that's it's in the same folder as our semester 1 slides. So you can always add in any feedback, any suggestions, what you found good, what you felt wasn't good, as well as anything really. Memes, I don't mind. Meme us.
Oh yeah, send us some memes. Like, we want your best memes.
Yeah.
Honestly, like, this is so chill. It's not like a very formal kind of setting. We're just kind of teaching you content, and then like you guys just like give us feedback.
Yeah. Especially if you run into any problems throughout the week, like a bad lecturer or technical difficulties that are unsustainable, do let us know, because we'll try cover that stuff in more detail. So yeah, that's all I have to say, I think. Do you guys have anything to add?
Just look after yourselves. Make sure you're washing your hands.
Keep the memes coming, because we love it.
Stay safe.
Everyone take time now.
Yeah, you guys try. We aren't going to get kicked out, but we do have to run to our tute, I think. So, in a few minutes. So we will see you guys hopefully next week. Message us with any of your questions, and we'll be happy to answer. But thank you guys so much for tuning in. Once again, you can watch the recording after this once it uploads. See you guys.
Bye.
Bye.